Hello student, welcome to digital classes. This video is for class 6. The subject is science and the chapter number 5, separation of substance. In the last video I told you about mixture, homogeneous mixture and heterogeneous mixture, pure and impure substances, purpose of separating the constituent of a mixture to remove harmful or undesirable like this, and method for separating the constituent of mixture by hand picking, sieving, winnowing, threshing, etc. Now today I am going to tell you about separating insoluble solid from liquid by sedimentation, decantation, loading, filtration, churning, etc. Now the separating of substance, this is the fifth chapter and this is the second part. Sedimentation and decantation is generally used for separating coarse particle of a solid from a liquid which are insoluble in liquid. For example, the coarse particle of a sand in muddy water can be separated by decantation. Before cooking, rice, pulses, etc. are washed with water. The washed water is removed by decantation. In sedimentation, we allow the sediments to settle at the bottom. Means we keep the beaker undisturbed for some time. Then the sediments are settled at the bottom because of a heaviness. Means the sediments are heavy, so it settled at the bottom after some time. Then in the second, you see we slowly pour the upper layer of the water in next beaker. This is called decantation. Separating a mixture of sand and water using sedimentation and decantation. In the second case, we pour the upper liquid, that is clear water, into the another beaker. This is called decantation. The process of separating insoluble solid suspended in a liquid by allowing them to settle down is called sedimentation. The solid particles that settle down during sedimentation are called sediments. The process of pouring out the clear upper liquid without disturbing the sediment are called sedimentation. Now let us perform an activity. Aim to observe cleaning cleaning of rice by sedimentation and decantation. Material required: a cup of rice and a bowl of water. Method: observe the rice and record your observation. Does the rice look dirty? Now mix the rice with water in a bowl and allow it to stand for some time. Without disturbing the layer of rice, descend the water. Observation: when the mixture is allowed to stand, most of the impurity come up and float near the surface, whereas the rice settle down or descend the water. Whereas the rice settled down on descending the water, we got clear rice. Conclusion: rice can be cleaned by sedimentation and decantation process. Now there is an activity in our book to obtain the clear water from the muddy water by sedimentation and decantation. Now the procedure: take small quantity of muddy water or a mixture of sand and water in a beaker. Allow it to stand for some time. The particle of the sand settle down to the bottom of the beaker. This settling down of of heavier particle is called sedimentation. Transfer the clear supernatant water upper layer of the water carefully to another beaker. This process of separating clear liquid without disturbing the settled solid particle is called decantation. Is called sedimentation. Now loading. In the first beaker, you see the muddy water, and alum is placed or dropped in that muddy water to settle the heavier particle faster in the beaker. Loading is a process by which the mixture of liquids containing tiny impurities is sustained by adding a chemical that stick to the impurity and make them heavier. For example, if we take a slightly muddy sample and add some alum to it, after some time we observe that a layer of mud settles at the bottom of the vessel and the water becomes clear. Here the alum got adhered to the small bird particles and made it heavier, thus causing the mud to settle at the bottom. This process is used during the filtration of water in the water filtration plant or a water filtration tank that supplies water to our city, town, etc. During sedimentation, the heavier porous particles settle down quickly. Fine particles of clay settle down very slowly. The finer particles can be made to settle faster by dissolving a small quantity of alum in muddy water. This method is called loading. Filtration. You see, this is a glass funnel. Filtered paper is put inside the or placed inside the glass funnel. When we filter any substance, the solid residue is left in the filter paper and the filtered water collected in the beaker, which is at the bottom. Now see, this is the setup for the laboratory filtration. There is a stand funnel and one beaker in which the clear water can be collected. Now the filtration. A mixture of an insoluble solid and a liquid can be separated by filtration, process in which the mixture is poured through the filter paper in a funnel and the residue is collected inside the filter paper. See the filtration. The method is used for separating fine insoluble particles from the liquid. In this method, the mixture is allowed to pass through a filter. A filter is a medium with fine pores. The insoluble solid particles are retained by the filter and the clear liquid is collected as the filtrate. Commonly used filters are a piece of muslin cloth or a filter paper, a layer of cotton or a layer of bed sand can also be used as a filter. Nowadays, special filters are used to obtain high purity drinking water. In filtration, the size of the pore in the filter is very important. For a good filtration, the pores in the filter should be or must be smaller than the size of a particle to be removed. Setting up of a filter paper in the funnel is shown. Filtering tea by using a strain is also an example of filtration. Here the tea leaves are retained by the strainer. The tea strainers cannot be used for filtering tur- turbid water. Means the tea leaves are slightly bigger particles, so with the help of strainer we filter the tea leaves. But if we want to filter the other thing in which the particles are smaller, then we are not able to filter that substance. So filtering of different substance, we use different filters. So that is why the pores in the filter should be important or must be important. If the particle size is smaller, then we use the small pore stainer. Now, churning. This method is used for separating lighter particles of a solid suspended in a liquid. For example, butter can be obtained by churning curd, obtaining from full fat milk. Cream can be obtained from unboiled milk by churning. Churning can be done by hand using a stirrer or by using a mixture commonly called mixie. In the past, you see in the figure, this article is used for separating butter from milk at home. Nowadays, we can use mixer grinder for taking out butter from the milk. Churning of cream. Churning is initially by agitating of cream causing incorporation of numerous air bubbles into the cream. When incorporate of air, there is increase in the volume of cream and air plasma interface. Surface active such as frictional impact, conclusion, etc. causes partial disruption of the globus membrane. The fat film thus formed serve as a foam depressant causing the air bubbles to burst. The liquid fat also serves as cementing material causing fat globes to clump together and eventually butter grains are formed which floats in plasma, that is butter. See, centrifugation is same like churning, but in churning the lighter component come upward, but in centrifugation the heavier components settle to the axis, like in the mixer grinder. When we use the mixer grinder, the heavier component rotate its axis. So centrifugation is a procedure that involves the use of centrifugal force of the sedimentation of mixture with a centrifuge used in laboratory and in laboratory setting. More dense component of the mixture move away from the axis of the centrifuge, while less dense component of the mixture move towards the axis. Now the application of centrifugation. There are two techniques on which the application of centrifuge is based on. Preparative technique, quite straightforward, consisting of placing the sample in a tube in the rotor and spinning the sample for a fixed period. Sample is removed and the two phases, pellet and the supernatant, may be separated by carefully decantation. Separate particles ranging in size from coarse precipitates to the cellular organelles. Second, analytical technique. In this, mostly measurements are taken to determine molecular weight, density, and purity of the sample. All analytical techniques required use of ultracentrifuge and can be classified as differential centrifugation, density gradient centrifugation. Basically, this centrifugation method is used to test blood. Now see what is centrifuge used for. Centrifuges are used in various laboratory to separate fluids, gases or liquid based on density. In research clinical laboratories, centrifuges are often used for cell, organelles, virus, protein, and nucleic acid purification. An example of centrifuge use in a clinical setting for the separation of whole blood component. Different assays necessitate serum or plasma which may be obtained with centrifugation. Now let us perform an activity. This is the activity number 3 which is given in our book, and this activity is related to the evaporation. To recover salt from its solution in water. Material required: china dish, iron stand, tripod stand, wire gauge, burner, salt solution. Heat about means the procedure. Heat about 20 ml of common salt solution in a china dish. When the water starts evaporating rapidly, lower the flame. What do you see at the end? All the water in the china dish gets evaporated and the white patch is formed on inside the china dish. Result: a white residue is left behind in the china dish. This is a common solid. This is a solid common salt. Conclusion: common salt can be removed from its solution by evaporation. We can get salt from the ocean water by this process. Means we get salt from the ocean water by the process called evaporation. Now I think whatever I taught in this video, everyone would understand. If there is a problem, I think there is no problem. If maybe there is problem, you will discuss within a class to clear your doubts.